And bonjour. This is Bruno LARVOL and Judith Perez here at LARVOL talking today about Pseudo-IPD in LARVOL CLIN, our platform for cancer data. Pseudo-IPD, how do we generate Pseudo-IPD in a way that is very novel, and I don't think anybody has the tool that Judith developed and is going to demo in a second. Before that, I've got a little infomercial, so to speak, for a book I just received last night. I haven't read it yet, from Reid Hoffman, the founder of LinkedIn, but also a big AI proponent. He has an AI company that is just starting in health and cancer oncology. That's our interest. And my friend Neil Peretz shared it with me. It's an advanced copy. It's not available yet, but I wanted to give it a shout out. I think it's going to be super interesting. It got a lot of testimonial from very well-respected people, including Bill Gates. So AI is going to change our life, and part of AI, of course, is data. And at LARVOL, what we're focusing on is cancer data. That's LARVOL CLIN. LARVOL CLIN is the Wikipedia / Bloomberg / AI company for cancer data. And Judith has built with her team a very exciting feature. Darko Medin and others worked on this as well. And I'd like you, Judith, to present that feature and give us a quick demo.
Yes, of course. Thank you very much, Bruno. So yes, we have a new tab, new introduction, new feature has come up into LARVOL CLIN, our web platform. We have worked with Darko, as Bruno has said, to build that new feature. And basically, the idea would be to be able to have that pseudo individual patient data, so we call it out pseudo-IPD data, available at each graph level. But let me walk you through it. So let's see an example. Let's make a search. So let's say, for example, HER2-positive breast cancer in here, and we will see how the feature works. And let's filter out also by ADCs.
Actually, just before you give that, I want to say one more comment where the data is coming from. So what we did is that we digitized all of that data from about 50,000 clinical trials, and we extracted the survival curves and also what's called the at-risk table. And then we used a biostatistical method called the Liu method to extract what is probably the largest private repository of pseudo-IPD. I believe we have 3.5 million patients, and it's growing very fast. And so what you're going to demo here is how the platform can use that pseudo individual patient data to create new analytics and help make decisions, both in research and maybe even in clinical practice.
Correct. So indeed, like in this dashboard, as Bruno is pointing out, we have the list of trials we have available in CLIN that match this initial search. And we have listed out, for example, for this immediate trial, this graph. So what we have done in here, it's, hi, Darko. Indeed, in this graph, we have two different KM curves, the trastuzumab emtansine one and the lapatinib capecitabine. Okay, so basically these curves have been digitized, as Bruno was saying, and with the coordinates as well as the at-risk data that goes along with that initial curve, plus the end size of those curves, we have been able to extract the pseudo-IPD data, so the pseudo-individual patient data, that we are now going to be using to create a pooled curve that is a combination of those initial two curves. So let's try it out. I'm going to be selecting for Emilia for this OS data in here. So we have the OS with trastuzumab emtansine. And let me check out also then Theresa, let's say, OS trastuzumab emtansine data. So once we have selected the curves, the idea is that the pseudo-IPD data from those digitized curves will be used to create a single curve, making a survival analysis from there. So if you check it out, this is the pooled curve that we have created based on our selection. Not only that, but we can check out which are the original curves that have been used to make that pooled curve. So in here we see that the median is 26, and if we check out the original curves, we have on top the Emilia one, and at the bottom the Teresa one. And if you can see the pooled curve, it's laying in between both of them. Indeed, they have similar sample size, a bit more for Emilia, but you can check it out, the pooled curve that has been created out of those two initial selections.
Some people, Judith, are calling this synthetic data. Some people are calling it, and we do, pool data. Dr. Ludovic Trincard, one of our friends and advisor, prefers that term. So we right now using the term pool data, simulated data. And it's not just the middle point between these two curves, by the way. The statistics behind it are very are more advanced than this. And this is why even if the bottom curve drops all of a sudden, statistically speaking, it doesn't mean too much. And it barely impacts actually the pool data. It's for someone who's not a biostatistician like myself, it's the first time it's a little bit shocking, but it actually makes sense when I was explained the very validated biostatistical principles and software used behind that pool data.
Yes, indeed. And now that you said it, when it comes to, for example, imagine we had one of the curves that were only 40 patients, the driving force would have been the other one, because as Bruno was mentioning, we have a statistical analysis behind it to support all the outcome. And not only that, but indeed, if we were to evaluate which is the accuracy of that curve that it's coming from the Pseudo-IPD data, we could even select just a single curve and let's create like a fake pooled analysis. But out of the, so in here we have selected just one curve. And if we show the original curve, it's lying on top of the pooled one, meaning that when we go from initial curve coordinates to Pseudo-IPD data and back to the survival analysis, our accuracy right now is really high.
Now, I've got a comment about that as well. And by the way, if you're listening to us, please drop us a comment, ask us a question. We love engagement. Also, follow us and subscribe, like to our content, so we can know that what we do matters and is appreciated, and we can produce more content. So please do that as we speak. The first time I saw that, I did not realize how important that was. I thought, okay, that's one curve overlapping on the other. Well, but it's actually remarkably important and impressive. Because what we did is that we really extracted not just the it's not just like we OCR the curve and replicated. That would not be very impressive. We OCR the curve. We extracted the patient behind the curve with additional data, and then we recreated. Okay, it's a little bit like I don't know, cloning a cat and putting one. I know Judith, that's a bad example. Judith has a cat in the background there. We cloned the cat. We put it in front of Judith and said, well, that's the same cat. No, it's a cloned cat. I said, well, it's not that impressive. Yes, it's very impressive. So that's a cloned, reconstructed via pseudo individual patient data curve, and it is beautiful.
Certainly. And there are many use cases that can be applied to this type of analysis. So if we were to be interested in how an MOA is working or in a particular condition, as we were checking out before on ADCs, or even then lay out different arms in there. So let's.
I've got one general comment unrelated to the data, but I want to make it as well. So 2 days ago it was World Cancer Day, right? And obviously at LARVOL we care very much about cancer patients, but we work with data. Oncologists who we really celebrate every day are the one on the front line, and pharmaceutical firms developing new drugs. We are on the data side. And when we're on the data side, it's easy to forget that behind every drop in this curve is a patient who either dies or progresses in his or her disease. And so I always want to remind that when we look at this curve to be a little bit um, maybe not somber, but serious for a minute. These are people. These are people, and so we're doing our part with our partner to help the field of oncology. But there's a certain seriousness that comes with these survival curves.
That's a very good point. Thank you. So maybe we could check out other examples if that would be nice.
Yes. And as you do that, I've got another comment. I know you're working on additional features, and maybe you can say a few words on that at the end as well. What is next for a pseudo-IPD feature? Because this is built in public. So built in public is like we share with you as it happens. Sometimes we make decisions even as we speak. Sometimes we find bugs as we speak or problems that we have to fix. And one of the decision we have to make, for example, is whether or not we merge the tab that says pseudo-IPD with the Kaplan-Meier tab in the middle there on the screen, which is the survival curve. Because really, survival curves and pooled IPD are very close in the way we use them. So we're considering merging the two tabs into one feature, one functionality.
Correct. That's like one of the steps that we are considering. And as Bruno was saying, also one of the interesting points or use cases for this pooled curve, like in here, I'm selecting Keynote 839 and Javelin Bladder 100 for urothelial cancer. And when you check out the original curves, they lie like really close one to the other. They are using different types of treatments. And the interesting point in here would be even to, like in a V2, in the next version of this tool, would be to even be able to overlay the control curve. So would be the counterpart of the treatment, right? So having the best supportive care, not having as an isolated curve, but on top of the previous one. So let me select it. But we have it in separate right now.
It's particularly valuable because when we talk with our pharmaceutical customers, people who are dealing with cancer data in pharmaceutical companies and are using them to better design their phase 3 cancer clinical trial, a trial that easily costs $100 million. Often what they need to do is to benchmark the standard of care, to understand what is the standard of care for that particular tumor type and setting and staging and so on. And to do that, they need to look at all the clinical data available. And this tool is, and they're doing it internally sometimes, but this tool is going to make it even easier for them. Their goal is to say, okay, for that tumor type, what is the expected survival curve if we do a clinical trial? And so how much better do we need to be compared with the standard of care, which often, actually, which nearly always should be the comparison. So they're going to compare themselves. There are going to be two lines in their clinical trial: the standard of care, sometimes chemo, sometimes another drug, sometimes, but very rarely placebo, and then the drug tested or the regimen tested. And what they want is a big separation, obviously, between the two curves, measured ultimately by a few methods, but the hazard ratio is the main one. But so they want to understand how will the standard of care perform in that setting and staging of these of that tumor type. And now they can.
Exactly. That's a really interesting feature that will be coming up. So to have the control and experimental arm there. So the really use case. Yes. Even also being able to download the coordinates of that pooled curve, that will be also a new feature that will be coming up under the Pseudo-IPD.
So I know we're getting at the end of our 15 minutes of build in public with LARVOL CLIN, and I want to encourage all the listeners to again like our content and subscribe to it, follow us, follow Judith as well, and follow me. And on top of that, I would like them to reach out to us if they would like either a demo of our platform, a demo of our Pseudo-IPD feature or other features of LARVOL CLIN, and potentially doing a pilot of our platform as well. There is a free version for non-commercial use, and of course a paid version for commercial use. So thank you, everybody. Judith, any final words before we close this Build in Public episode?
I'd say let's give it a try to that Pseudo-IPD. I would like to hear like the thoughts from our viewers, of course. Thank you very much.
Au revoir.